Okay, let's go ahead then. I am Joanne Geer. I am the Executive Director for the Biopharma Research Council, and I want to welcome everyone to the fourth session of our short course in extracellular vesicles. I want to give a great appreciation to our organizers and partners and sponsors. Could we have the next slide, please? That's me. I'm going to go into the next one. We're going to do a very short poll just to get a sense of who is in the audience, and this will give our speakers a little guidance on who they're talking with. So you can go ahead and submit your answers right now. The Biopharma Research Council is essentially an association for scientists throughout the biopharma research community. Industry, academia, nonprofit, government, and supplier laboratories, and I like to say, and those who love them. And a lot of what we do is get scientists and support folks together across as many different silos as possible, essentially to learn from each other. We do a number of webinars. I'll share with you some of our upcoming webinars in a moment, as well as symposia, conferences, and roundtables throughout the year. I think we're just about ready. I'm going to close the poll. I think we'll all see the results, but I'm going to read them off just in case. So we have 23% who are just getting started with the exosome, 45% who've been at it for a while, 27% who are interested, and who might get involved, and 5% who are just interested. There you go. Thank you very much. Terrific. So we can go on to the next slide. Our next event is going to be in North Carolina at Research Triangle Park. This is a really interesting full-day program of across the community. This includes agricultural sciences as well as biological sciences, as well as nanotechnology and all kinds of other really interesting breakthroughs that have gone on in the past year. Go on to the next slide. Our next webinar is going to be on November 20th. We're doing kind of a roundup of different ideas that have been emerging this year. The Mobile Patient is a series we've been doing starting this summer, an occasional series, and we'll be doing a program on emerging FDA guidance related to devices intended for the home. This is a really wonderful program. This is the 6th time we're doing an executive retreat that's all about preclinical and actually early clinical outsourcing. And we have some wonderful co-chairs, and we'll be hearing some more about that. That'll be in the Princeton area. You can enter a question at any time. We will probably take all of our Q&A at the end of both presentations today, but you can feel free to enter at any time. There's a question box on the side of your console, and a question I always get asked is, will this be available later? And the answer is yes. The recording will be available on our website as well as the slides. Sometimes the same day, sometimes within a day or two, and we send out an email to let you know that they're available. And we just want to thank everyone who's been involved with setting up this series, beginning with, go to the next slide, please. So Winston Kuo, who, if you've been at the other sessions, we had a much more serious picture, and then I saw that Winston had this wonderful picture of himself at Mount Rushmore. So I think, you know, he deserves to be up there with all those great guys. But he's definitely put this all together, and you'll hear more from him in a moment. Just to review what our series has been, and all of the previous sessions are on our website. Go ahead, please. We have the next slide, please. We started with 4 weeks ago with Jan Lotval, who is one of the primary researchers in this area. Thank you, from University of Gothenburg. And then Johan Skog, who you can just go right through these slides. Thank you. From Exosome Diagnostics, who talked about the applications. followed by Sasha Vlasov speaking about the commercialization opportunities. And finally, today we're going to hear from Eva Rhoda and Angel Sayuda about the funding and regulatory issues. I'm going to tell you a little bit about Winston, and then you can go right through these, right up to the next slide of Winston. I'm going to and so Winston Kuo is the COO of IES Diagnostics, developing diagnostics based on a technology licensed from the FDA and focusing on systemic lupus erythematosus. He's also the editor-in-chief and founder of the Journal of Circulating Biomarkers. He sits on the scientific advisory board of Exosomes and Hansa Biomed, both focused on exosome-based diagnostics and life sciences products. His research interest in extracellular vesicles includes the technical aspect of commercializing, both from life science and clinical aspects. Winston.
Hello, everyone. Thank you, Joanne, for the introduction. I'm delighted to host the fourth session in collaboration with the Biopharma Research Council and Intech Publishers to provide the short course on exciting area of extracellular vesicles, the transition from tissues to liquid biopsies. At this moment, I would like to take to let everyone know about the Journal of Circulating Biomarkers. We encourage you to submit to our journal free of charge for original research, manuscripts, reviews, perspectives, opinions, whether it's technical or any material of your choice. And please email me if you should have any questions. So today we're really excited to have a panel in which Joanne had mentioned, made of Eva Rhoda and Angel Sayuda. As you heard, we will hear about the regulatory aspect of biological medicine and advanced therapeutic products as it relates to extracellular vesicles, and then also about international funding opportunities. And all the webinars were outstanding and very informative, and they are accessible on the website with their talk and presentation. Make sure you click on the link. So I'm going to jump right into why is it important. I mean, extracellular vesicles, including exosomes, macrovesicles, have shown to carry a variety of material like mRNA, proteins, microRNA, other nucleic acids, lipids, and within the past 5 years, they really have emerged as promising, minimally invasive novel source of material for molecular and proteomic diagnostics. So although extracellular vesicles can be easily identified and collected from biological fluids, further research and proper validation and standardization is definitely needed in order to bring them to the clinical setting. So today, we will definitely hear a different aspect from what we heard before earlier in the 3 sessions, really the regulatory aspects of extracellular vesicles, and then also how to sort of get your hands onto additional funding to support your initiatives. So today we conclude the short course with the panel discussion, and just a few housekeeping items again. As the format of the panels is as follows. Each speaker will have about 17 minutes of time to present for a total of about 35 minutes for the presentations, and after which we will open it up to the audience, the participants, for question and answers once the 2 presenters have presented. But please submit your questions during the presentation by entering them on the dashboard to the right of your screen. So our first panelist is Dr. Eva Rhoda. She is actually currently the head of the Transfusion Medicine Department and director of the GMP laboratories at the Panelists Medical University in Salzburg, Austria. So Dr. Rhoda made her medical education at Karl Franzens University in Graz, Austria. She finished her training in transfusion medicine in 2005 and worked as a postdoc at the Stem Cell Research Unit of the Medical University of Graz. Her research activities focus on cellular therapy and regenerative medicine, especially on the human mesenchymal, stromal, and endothelial progenitor cells from various tissues and the manufacturing of cell-based therapeutics. The postgraduate training in pharmaceutical quality management enabled her to act as a qualified person for the pharmaceutical manufacturing of blood, cells, and tissues. Since 2013, she was a member of the European Cost Action, MedHod BM1201, Macrovesicles and Exosomes in Health and Disease. Currently, she's establishing a GMP Laboratory at Paracelsus Medical University in Salzburg for the production of human advanced therapy medicinal products and or cell-based pharmaceuticals such as extracellular vesicles. Thank you very much, and now we'll hand it to our first panelist, Dr. Eva Rode. Please enjoy.
Hi everybody. Good morning USA and good evening in Europe. Hi everybody, my name is Eva Rode and I will now go to tell you something about our issues and pharmaceutical engineering aspects of extracellular vesicle-based therapeutics, above all also of our issues bringing together regulation and biological complexity in the concepts of developing new investigational new drugs. How did it all start? From 2001 to now, my work was more based on routine blood production, patient blood supply, diagnostics and therapy, but always already related to the establishing of cell-based therapies. We tried to focus on novel cell-based therapeutics for tissue regeneration and maybe also to achieve functional repair after organ injury. In the development of emerging investigational new drugs which are based on human cells or derivatives such as extracellular vesicles, you face several hurdles and requirements which may hamper development or also velocity of your experiments and all the problems you can face with the general biomedical complexity if you want to understand tissue regeneration. There are many open questions, but above all, other problems come along, like you need the adequate infrastructure. There is the need for a quality management system to implement manufacturing procedures according really to pharmaceutical standards in order to generate medical products which can be tested in clinical trials. And this is not enough. You also have to face complicated regulatory legal frameworks, and with regard to vesicle-based therapeutics, up to now it's not yet clear if the pharmaceutical categorization is really finally defined. This is a challenging question and international harmonization is lacking. So with regard to let's see, I have here, with regard to the regulatory aspects, the 3 points, the infrastructure management system and regulatory issues are those we are dealing with. And of course, there are open questions regarding biomedical mechanisms. Now I have to, okay. Here you see an example where we try to establish the production of mesenchymal stromal progenitor cells in the in a GMP environment. And as you can see, in the human setting, you have other dimensions as compared to mouse experimental settings. You need liters of culture media, you have pellets big as golf balls, you need a whole incubator for one person to get about 1 billion cells, and you have square meters of cell culture dishes. In this case, we expanded mesenchymal cells from bone marrow aspirations, and we wanted to produce advanced therapy medical products, which are also called ATMPs, and these are especially regulated with regard to pharmaceutical production and clinical testing, as you will hear later on. But these MSPCs can also be the source to generate extracellular vesicles. And that's why I'm showing you this example, because here you can see that we have about 6 liters of supernatant in 10 of these culture dishes, and there you can really generate sufficient quantity of vesicles out of culture media, if you want. No, it's not okay. Another important issue is to avoid xenogenic factors out of human cell culture if you want to expand cells in order to apply them in clinical trials. A colleague of mine, Katharina Schallmoser, she is really since years working on the studying of the impact of platelets on regeneration. And as we are a blood producing institution, we can take out of whole blood donations or from single platelet donors the platelet concentrates, pool them and make pooled human platelet lysates and apply them for cell culture and to avoid fetal bovine serum. This is one of a compassionate use experiments we had with not an experiment, but a clinical application in a critical size bone defect of a patient who really suffered more than 10 years of this 10 centimeter bone defect in the tibia. After 1.5 years, this patient got rid of all his implants. So this means, together with these mesenchymal progenitor cells and pooled human platelet lysate, we could achieve bone regeneration in this case. We could also then repeat this experience with another patient. Selected publications, we don't go through these slides, busy slides, but they are with relation to the potency of mesenchymal cells or also endothelial progenitor cells and towards regeneration, and also they are focusing on the GMP compliance of production processes. This led us then to the privileged situation that we got funding for a tissue regeneration center in Salzburg that is mostly focused on spinal cord injury, but also to other tissues for regenerative therapies. Spinal cord injury is not my core competence, and I was happy not to be alone here with the expertise, because sponsors, they gave us the mandate to focus also on neuro regeneration, which is okay. And we got the mandate to do clinical research related on spinal cord injury, but also to develop novel integrative therapeutic strategies for tissue regeneration and functional repair after organ injury, not only related to spinal cord injury. In this integrative approach, we try really to cover basic research and translation, as well as application to patients with standard of care, and to combine all this together with clinical and basic scientists. And my part in this consortium is focused on the development of cell and cell-based therapies. I already told you I was happy to find that a local expertise here was already existing with regard to neuro regeneration and clinical neurology. Also with regard to vessel regeneration, and also from retina regeneration out of the ophthalmology department, skin regeneration, pelvic floor, bone regeneration, and also with vessel. and endothelial progenitor cell biology. So we could really do a kind of panel of experts who focus on similar but not really totally the same goals. And we really organized that clinical wards work together with scientific preclinical units, have a platform with core facilities, including this GMP laboratory, really to cover the translational processes which are needed for these interactive efforts we want to organize and to make then some therapies real. For instance, stem cell therapies or other cell-derived biologicals for regenerative therapy. So based on the assumption that there can be regeneration after the different organ injuries by inherent healing power of stem cells or also factors, we went further and asked what can we do for spinal cord injuries or tissues and organs. And there really the biomedical complexity gives you many open questions which are not yet solved. And as we then met Jan Ledwall and read literature, we also found out that extracellular vesicles might be a very innovative source for doing clinical research. We know regarding regulations, we really have to think about the questions, how can we prove identity, purity, or potency of such biological multimeric complexes like it is with extracellular vesicles. So we have to address the questions when we characterize EVs. What are the isolation techniques? How can we quantify them? How can we make purity versus impurities? How can we prove purity? The next is, I don't go through the busy slides, but what are the questions? We need to know mode of action of these vesicles. How can we really prove this? How can we find out this? What is the nature of the target which is modulated by EVs? What are the right potency assays in vivo or in vitro? And how relevant are the selected animal models we want to test with regard to the intended therapeutic use? Next questions are, what about vesicle uptake, vesicle homeostasis? If you apply them systemically, what is the biodistribution? How can we cover with non-clinical and clinical safety testings, as you have to do with every biological medicinal product you develop? What about safety pharmacology studies? Are they needed if you use vesicles for an autologous use? Or can you avoid some of these very laborious steps towards the development of a clinical applicable pharmaceutical product? This is biologic complexity. Now we have something which is much easier if you get funding. With regard to the infrastructure, we finalized construction work for a research center in the November 2013, and we really could, right from the scratch, design and establish a GMP laboratory, which now has been already implemented. So the infrastructural part itself is somehow costly and laborious, but it's feasible. Same is valid for the quality management system. You need it for manufacturing of each drug, especially investigational new drugs. If you really want to go into clinical testing, you need to implement every step with the help of good manufacturing practice, which is not a enigmatic thing, but it has to be done, and it's very cost and labor intensive. But for the development of, say, cell-based or vesicle-based biological medicinal products, you need these principles, and you have to produce products in line with a stringent quality management system. As with regard to hurdles we have faced the last years and also for the future, we think that open questions in the biomedical complexity still remain, and the complicated situation regarding regulatory issues on EV-based therapeutics is also still remaining, because we now need to get an idea what is the pharmaceutical categorization of vesicles, and is there already international harmonization between Europe, United States, Asia, Africa, Australia? I have not the impression that this is very harmonized up to now. I'm now apologizing for the next busy slides, but I cannot somehow, yeah, you have to face it, because it's really the current legislation. It does not provide specific regulation on EV-based therapies, and so my personal impression, and I also confirmed it with our authorities, is that now EV-based therapeutics are biological medicinal products. But each of you who is not from Europe or from, especially from Austria, should contact his own national authorities to confirm this information. This has a special implication, as EVs are classified as biological medicine, which contain one or more active substances that are made or derived from biological cells. Some of them may be already present in the human body, such as erythropoietin or insulin or growth hormones. This is valid for extracellular vesicles also. The active substances, maybe they are not yet known, but they are for sure larger and more complex than those of non-biological medicines, and only living cells and organisms are able to reproduce this complexity. This pharmaceutical classification as biopharmaceutical, as biological, it harbors special challenges with regard to the manufacturing and the preclinical safety testing. But it, up to now, and this is my information, it has to be done in parallel to other biological medicine. With regard to the source of EVs, it could be out from cells, from body fluids, from tissues. For instance, I read one clinical trials where ascites-derived EVs were used against colorectal cancer. So you can use it also from non-expanded cells. But already minor changes in your production process can have a major impact on the clinical characteristics, and therefore the manufacturer, they may face great challenges in the complete definition of physical, chemical, immunochemical, and biological properties of extracellular vesicles. And for the characterization of this biological medicinal product, it has not only the product that you have to test, but you have to go for a combined approach and test all your expected active substances. This can be one. I don't expect that with vesicles it's only one active substance. You go for safety pharmacology, pharmacodynamic, and toxicology testing, but you also have to test not only the final medicinal product, but you have to really test and assess your production process and your controls. You cannot skip this, because the production process itself may alter the product which is generated in a manner that may harbor patients. Let's see. And there could be a good way out of this dilemma that it's really difficult to characterize your vesicles in a tight and controlled manner. We could get hospital exemption rule as it is defined in the ATMP regulation in Europe, and as I heard also, it can be given by the FDA's Center of Biologics Evaluation and Research. So this means the ATMP regulation is valid for medicinal products, which are also biological medicine, but they are referred to gene therapy, cell therapy, or tissue engineered medicinal products. These products are prepared on a non-routine basis, and according to the normal and the specific quality standard, which means current GMP standard. And ATMPs have then to be used in the same member state in Europe, in a hospital under exclusive responsibility of a medical practitioner. And at least for the production process, it would fit for vesicle-based therapies. But the ATMP regulation itself is up to now not valid for vesicle-based therapies. It would relieve us from very, very tight preclinical and non-clinical safety testing, but it's not valid as defined by now, because vesicles are not ATMPs. They are classified as biologicals. ATMPs are defined to contain living cells. They should have a nucleus. That's not applicable for EVs. And they are not intended to be used for the same essential function in the recipient as in the donor. This is very bizarre, but it's true. For instance, hematopoietic stem cells, which are going to be transplanted and then they make hematopoietic reconstitution, make the same essential function. These are not ATMPs, even if they would be substantially manipulated. So having a nucleus is essential for the ATMP definition up to now. The ATMPs, they are not considered to be comparable with conventional medicine with regard to the physical, chemical, immunochemical, and biological properties. And this would be the exit strategy also for extracellular vesicles if we could apply the hospital exemption rule. We think that vesicle-based therapeutics are also not comparable with conventional pharmaceuticals, but we have really to convince the legal bodies, international legal bodies. It's generally acknowledged that conventional non-clinical pharmacology and toxicology studies may not be appropriate for the cell-based medicinal products, it has the ATMPs. This would really help us also for the development in the preclinical setting for the EV-based therapeutics too, because testing is not that strict as compared to conventional pharmaceuticals. Therefore, the preclinical safety testing to clinical application of ATMPs is really separately regulated by this hospital exemption rule, at least for the European countries and the United States. This can be valid. I have not figured out how it's how it's regulated in Asia, Australia, Africa. There are really varying national rules which are not yet harmonized. In conclusion, I think that the scientific community and also interested companies in vesicle research and interested in EV-based pharmaceutical development, they should contact the relevant national and international legal bodies to discuss issues on manufacturing and interventional strategies of EV-based INDs. The earlier the better. And possibly the hospital exemption rule can be achieved for EV-based INDs too, not only to ATMPs, especially if we harvest the EVs from cell products which are within the ATMP regulation, because they cannot do any more harm than this resource ATMP resources themselves. But we really have to discuss this with the authorities. Currently, EVs are classified as biological medicinal products. And I think here we need the scientific, biologic, and really pharmaceutical manufacturing arguments to convince our partners from national authorities and international legal bodies, because regulations are made by human beings. Thus, if they are quite complicated, we can this doesn't mean that we never may change them. In contrast, with regard to the biological mechanisms behind vesicle-mediated regulation, I think this can in fact be very challenging issue, which is not that easy to unravel. Here, it's the really big challenge behind. So we should really earlier we should go for discussion with competent authorities in order to convince them that we need the investigation of biological mechanisms which are behind vesicle-mediated regulation of tissue homeostasis and regeneration. This is only feasible if we overcome regular hurdles, and this should be of a very interest for all of us. Yeah. This is the transfusion medicine in Salzburg. We are more focused on the routine production, and each of us who are here in red are involved also in doing research. Some of our research colleagues have just started and we don't have any picture yet. And I thank you very much for your attention. And we thank all the sponsors who really support this exciting project we may now start in Salzburg, and we have already started. And now it's open for questions.
Well, Dr. Everode, thank you so much for such a wonderful, outstanding, and informative presentation. As we mentioned earlier, we'll hold off the questions and answers to the end.
I'm sorry. Yeah, I forgot.
It's okay. It's okay. But for those that are still online, please continue to submit your questions. So our next panelist is Dr. Angel Osoyo Cecido. He's the director of the Thoracic and Brain Tumor Laboratory in Madrid. He completed his PhD at Merck, Sharp, and Dome, and he worked as a postdoc fellow within the medicine department of Mount Sinai School of Medicine and the neurosurgery department at Cornell Medical Center. He went back to Spain and he co-founded the Glioblastoma Spanish Network. Currently, he's the director of the Thoracic and Brain Tumor Laboratory at the Hospital de Madrid Foundation, and the president of REG, which is the Glioblastoma Spanish Network. His research is focused on the use of extracellular vesicles as biomarkers for diagnostic and follow-up treatment, as well as for the evaluation of drug sensitivity in patients diagnosed with high-grade gliomas. So even though he has very extensive research and focus on various different aspects of extravesicles, I sort of put him on the spot and asked him to talk about the international funding opportunities. So thank you very much, and now I'll hand it over to our second panelist, Angel. Please enjoy.
Thank you very much, Dr. Kuo. And as Dr. Kuo introduced, I'm the director of a brain tumor laboratory. So the information I'm about to present about the European Union funding opportunity is based on my experience as an applicant. I'm not I don't have a deep knowledge on European Union bureaucracy, but I think still I can share some information that I hope is going to be very useful for all of you. I know that we are many countries in Europe. There are many institutions, and sometimes it's very difficult to get the big picture of how the European Union is organized. So, but here I would like to divide the presentation into three main blocks. The first one is just to give brief picture of how the European Union is organized, because it's very important to know how is it in terms of understand how is the financial support for research and innovation. Then we will pass to the second block, where we are going to describe a little bit how the Horizon 2020, that is the financial tools we have in Europe to support the research and innovation, and how it works and how it's organized and what kind of calls actually grant. And finally, we are going to end up with a description of partnership opportunities, as many of the calls are focused on funding coordinated projects. It's very important to get the proper partners, so we all can apply together with more guarantees. So once said that, in this slide you can see the three main institutions in the European Union. The first two here and here is the European Parliament and the European Council. They are more on the politics guy. They are all politicians, but these two institutions is more in charge of the law, regulation, and annual budget approval. Then we have this other one, which is more important for us, the European Commission. They are more like the technicians between the politicians, and it works pretty much like a national government. So we have a president, and then there are a few, I don't even know how many there are. They change every other time, but they are called the directorates in general, which actually works like a ministries. And the one that is important for us is the DG for research and innovation, as they manage the money, the budget for research and innovation funding. So how everything works? Well, in Europe we have like 10 years program. And every 10 years, the program changes. For every program, for every 10 years program, we design a strategy, then identify the goals, the objectives, and then design the financial tools, the financial instruments for different fields. In our case, for research and innovation. So the previous program, the strategy for the previous program was called Lisbon Strategy, and it covered from 2000 to 2010. And the financial instrument to achieve the goals in the fields of research and innovation was called the 6 and 7 framework, that probably you heard about, even those of you that are not living in Europe. But right now we are in a different program, in a different time. So the program is called the strategy for this program is called Europe 2020, and it covers from 2010 to 2020. So the financial instrument to support the research and innovation in order to achieve the goals is called Horizon 2020. So once we know where the Horizon 2020 came from, we can be focused on how is it organized. So the Horizon 2020 is organized in three main areas. So there is investment in other actions, but these three areas are the most relevant. In all three areas, excellent science, social challenges, and industrial leadership, we can find goal focus on projects at different levels of development, according to the technology readiness levels. So in excellent science, most of the calls are focused on projects at level 1 to 4, 5, social challenges from 4 to 7, and within industrial leadership, we can find call to support projects involving products that are actually about to be in the market. And all three, we can find calls that are suitable for microvesicles-based projects. So now we are going to the web page of the Horizon 2020, and I will show you how to look for a specific calls within all these three areas. We will see that in social challenges, we can find the majority of the calls that are suitable for microvesicles-based projects. So we are going to start with the Excellent Science, and here on the left, we can see the web page of Horizon 2020. So here we see these windows, and these windows scrolling up and down, we can go to from one to another area. If we click on Excellent Science and all the sub areas, this is what we see today. Well, actually yesterday, I did the research just yesterday, so we can everything up to date. Here we find all the calls that are open, but none of them are, of course, suitable for microvesicles projects. I just highlight these three that are all within the ERC program, the European Research Council program. There are three types of projects that we can apply within this program. One thing that is important to consider is that the application for these calls are individual. So individual researchers can actually apply for those grants. This is important because it's very different to the following two groups of calls. And also it's important to notice that the topic is free, so you can actually apply for everything you have in mind. Also, there are some areas of priority areas. Pretty much you can apply for every type of subject. Still, there are three main type of grant: starting grant, consolidator grant, and advanced grant. The starting grant is for people who defending their PhD thesis in the last 2 to 7 years. The consolidator grant from 7 to 12 years, and the advanced grant is for people with a very strong publication record, really with a name within the field. The starting grant you can apply for 1.5 million, consolidator grant for 2 million, and advanced grant for 2.5 million. This type of grant is pretty much like a R01 in USA, those supported by the NIH, and well, they are actually a very good grants here in Europe. And then you have this other one that is it's a bit less important. It's just very new to demonstrate proof of concept. To apply for this grant, you need to have previously one of these three. And you get to the point where you have a product about to get the market, and then you can apply for this one to demonstrate proof of concept. This is about 150,000 euros, not much. So let's go to the second main areas of Horizon 2020, which is industrial leadership. There is a big difference between this block of calls compared to the first one. And one of the main important differences is that it's a company which has to apply for the grant, and a small and medium company. And there are like three phases where the company can apply for demonstrated feasibility assessment, and they can apply for 50,000 euros. In a phase 2 innovation development from 500 to 2.5 million, and then in phase 3 for commercialization. That is more undefined, the amount of money you can apply for. I did also a research yesterday, clicking on industrial leadership and on all the sub area, and I just found 2 applications that might fit with microvesicles based projects, one in phase 1 and phase 2, and this is the actual call: clinical research for the validation of biomarkers. I think the microvesicle projects actually fit very well on this one. The only problem is that you need to associate to a company, and the company is the one who actually applied to the call. But the company can actually associate it with many different research groups. So then we go to the social challenges area, where we can find most of the calls that actually fit the microvesicles projects. I did also the research yesterday, and clicking on social challenges on health, we see these 3 calls that match perfectly with microvesicle-based projects. There is also a difference between these projects and the other 2 blocks of these calls and the other previous 2 blocks of calls. For this one, the applicants are groups of researchers. So we need 3 or more countries involved, so at least 3 groups from at least 3 different countries in the European Union. we can apply for something between 4 to 6 millions, sometimes even more if you are able to justify that. And basically there are two different types of applications. One is called one-step and two-step. One-step is you need to write down the whole proposal. And the two-step, you just write down a letter, about 15 pages more or less, and then if the referee agrees, they can invite you to submit the rest of the proposal. So actually, if eventually you are rejected, this saves you a lot of time. But the important thing is that between these modules of application of calls, we can find many different subjects where microvesicles-based project can actually fit, like new approach to improve predictive human-tested microvesicle for biomarkers, understanding disease, system medicine. This is more basic biology of microvesicles or biomarkers or any kind of things. We have this other, like new therapy for rare disease. We can actually we can apply with grants focus on using microvesicles as vehicles for drug delivery, for instance. So actually, there are many opportunities within these the social challenges. I want also to highlight that these are the calls that are actually open, and every 2 years new calls are open. So it's just a question of going back to this website and review everything every few months. So once we decide the calls we are going to apply for, one of most important thing is to get the partners. As you saw before, with the exception of the ERC, the rest of the application needs to be done by a group of researchers, so you need partners. And looking for partners, there are 3 main ways. The first one is looking for partners at websites, looking for specific profiles or specific interests. The second one is by setting up networks or scientists where you can meet other people with similar interests. And there are websites supported by the European Union that help to construct networks. And of course, the best one is the one that actually works better is direct contact. Just when you see someone that might be interesting by their publication or whatever, send an email or make a call, and that always works. In any case, this is the web page where you can actually find for a partner, and you can also publish your profile or your project interest. So waiting for someone to contact you directly for apply together. This is one of the network that has been running here in Europe. This is actually very complicated. ERANET what actually does, well, it's a program aimed at fostering the cooperation and coordination of research activities, and it's carried out at national or regional level. So it's kind of difficult to access to this network as an individual researcher. You have to contact your representative in your country. This is the network that actually works better. And actually, I think Eva was part of this network. This is called COST, the European Cooperation in Science and Technology. It's an institution different from the Horizon 2020, but right now they have some kind of agreement. And what it does is very easy to apply. When the call is open, you just submit a couple of pages showing your interest in a specific subject, and the people across the European Union that are also interested on that subject, and if the referees agree and open a time where many people can join the network, and then the network is set up, like this one that is already set up, and it's called European Network of Microvesicles and Exosomes in Health and Disease. It ran for 4 years, and they give you money just for traveling and organizing meeting and congress. They do not give money for research, just for networking. So this is pretty much just to sum up. Horizon 2020 is the financial tools to support the investment in research and innovation according to the strategy called Europe 2020. there is two groups of two blocks of course where we can apply with the subject of with microvesicles-based projects. One is the excellent science through the ERC, the industrial leadership with companies, and there are many calls open within the block of social challenges. There are also tools for looking for partnership for partners, and more important, if you want more information, more specific information, in every European country there is a national delegate or the national contact point. Sometimes they are the same person, sometimes they are not, but they are the person who actually even more details that you can find in the web. And I think I try to be brief, so we don't spend most of the time with the presentation. We can take a question. Winston, if you think we can go to questions and take some. Yeah. Okay.
Thanks.
Thank you very much.
Thank you, Angel. And also I want to thank Eva Rode for such a very informative and detailed presentation on both aspects. We're still, still continuing to accept questions in light of the time. If we do not have time, for today to answer your questions live, we will definitely provide a link with the seminar with the responses to questions posted. So please continue to submit your questions. I have a question for Eva. I'm actually also the editor-in-chief of another journal called Nanobiomedicine. And today there's so many array of nanotechnology products being applied to therapeutic medicine, like magnetic nanoparticles, quantum dots, liposomes, microspheres. So you can imagine. And while most of these technologies are still in preclinical development, however, there are a few FDA-approved liposomal agents in the market. How can we learn in reference to these things that are already in the market to accelerate the process for utilizing extracellular vesicles into the therapeutic space? And the second question I have is also, since you are in a process of establishing a GMP facility for perhaps manufacturing extracellular vesicles in the standardized ways, when do you expect to be able to sort of make these accessible for collaborators in the near future?
Okay, I'm getting first question first. When we are getting ready for external cooperations, with regard to our manufacturing license, we will be at least engaged for another 2 months or 3 months until we get the actual manufacturing license. Then we have already done much work in establishing processes for our own procedures, but I think I cannot say you an exact horizon, but it seems to me somehow I cannot say something between 1 and 5 years, which is really because of consolidation of all these things and really addressing the first clinical applications we want to intend. So I think it's necessary to stay in contact, because sometimes we cannot imagine the needs of other partners, and maybe it's not that complicated to get in cooperation. This is so. With respect to the license, I would say half a year, but with respect to our own capabilities, I cannot give you an answer.
Okay.
And for the how can we learn from liposomes or other nanos or quantum dots? Yeah. That's a good point. I would in the organization of non-clinical safety and then also clinical safety testing. It's in fact very intelligent to look for partners who have done all of these safety testings already with biomimetic or other liposomes or biomimetic vesicles, if there are already some in the world. But I guess there is much done in the other preclinical development of nano particles, and so there should be kind of networking to this. But I confess I have not yet contacted contact to any of them, because we are also newcomers.
Yes. Okay, thank you, Eva. Joanne, I'm not sure if we have enough time now. Actually, I see it's 12 o'clock for additional questions, or should we make them available to the participants later on?
Well, thank you very much, Winston, and thank you very, very much both to Eva and to Angel. What a really beautifully detailed and comprehensive presentations. It's pretty exciting for us to be part of this emerging world, and we appreciate your expertise very, very much. We will have the recording and the slides up within a day or so, and we'll be sending out an email. You can go to our website, which is biopharmaresearchcouncil.org. Thank you. And you'll find the recordings for the previous sessions as well. We've gotten such a great response from this series that it's very likely that we'll be exploring this content more. So feel free to be in touch with us if you have ideas, questions you'd like to see addressed, presentations you might want to share of what's going on in your own lab. We'd be very, very happy to hear from you. And Winston, this is the final session. Thank you for the moment. Thank you so much for pulling this together. You've invited fantastic speakers, and it's really been a pleasure working with you. So thank you, everyone.
Thank you.
Thank you. Bye,
Take care.
Angel. Bye.
Bye-bye.
Thank you. Bye.